Hi, I'm Naeha Subramanian and I'm an associate professor at ISB. Innate immunity is literally the form of immunity that we're born with. It's a first line of defense against pathogens or infectious agents, or for that matter, any foreign substance that enters into your body. It's composed of cells and tissues that essentially serve as barriers to entry of pathogens and foreign substances. This includes things like your skin, the mucosa, and the epithelia that line these mucosal surfaces. The cells and tissues of the innate immune system express molecules called pattern recognition receptors, or PRRs. These PRRs can sense and they can respond to a broad variety of infectious agents. Upon doing so, they respond by producing molecules called cytokines. You can essentially think of cytokines as a burglar alarm that goes off when an intruder enters into your house, which will set into motion a series of events to contain the situation. Similarly, sensing of pathogens by the innate immune system sets into motion or orchestrates a series of events that lead to recruitment of different types of cells, which have different types of functions, and instructs the development of a more specific type of immunity called adaptive immunity. Now, the innate immune system is a double-edged sword. So there's a catch here. While the innate immune system is absolutely required for host defense against pathogens, if it's dysregulated, that is, if there's too little or too much of it being activated, then it can lead to a wide variety of diseases. So things like autoinflammation, susceptibility to infections. It can also set the stage for certain types of autoimmunity and cancers. Currently, we're working in three areas: host-pathogen interactions, the basic biology of NOD-like receptors, which are a type of pattern recognition receptors, and Lyme disease. In the context of host-pathogen interactions, we've been really interested in this arms race between our immune system and between pathogens. And we're focusing primarily on the study of intracellular bacteria, such as Salmonella, which causes food poisoning, and Mycobacterium, which causes tuberculosis. These bacteria have actually evolved to hijack the normal machinery of our cells, so hijack our innate immune systems, and they've learned to survive inside the really hostile innate immune environment of a certain type of cell called the macrophage. Salmonella, for example, has actually evolved to co-opt our innate immune response and conceal itself inside macrophages. And we think that future work in this area has the potential to impact development of host-directed therapies and strategies to tackle antimicrobial resistance for these intracellular pathogens. The second area we're working with is on the basic biology of NOD-like receptors, or NLRs for short. NOD-like receptors are actually the largest class of pattern recognition receptors that detect pathogens in danger. In humans, there are about 23 NLRs, and mutations and polymorphisms in them are associated with a wide variety of diseases. But we don't completely understand the immunological functions for many NLRs and how their dysfunction can be linked to initiation of different disease cascades. So for instance, we do not understand their signaling pathways, how these signaling pathways are regulated, and how their dysregulation is coupled to disease initiation. And so using systems biology approaches, we are trying to decode NLR function and its regulation. We are trying to uncover what the normal immune functions of these NLRs are and to reveal their links to disease. And finally, the third area that we're working on is Lyme disease. This is a disease caused by the pathogen Borrelia burgdorferi, which is transmitted by the bite of ticks to humans. This typically results in a rash called a bullseye rash at the site of infection. We are interested in the heterogeneity of the immune response to Borrelia infection. different people manifest very different types of symptoms and have different trajectories over the course of the disease. One outstanding question in the field is why some people resolve disease with antibiotics while others progress to post-treatment disease. And so using systems biology approaches, we are collecting information at multiple scales and we are integrating these data to derive mechanisms of pathogen clearance and to derive immune correlates of disease. So Lyme disease has actually seen an increased geographical distribution worldwide over the past decade or so. And currently it's believed that there are about 300,000 cases in the US each year. The annual economic impact in 2019 alone was close to 786 million. The societal impact of the disease is actually likely much larger, but it's not completely studied. One of the problems associated with Lyme disease is diagnosis, because current diagnostic tests remain negative for weeks after infection, which means that some people never get diagnosed or get improperly diagnosed. There is also currently no vaccine available for the disease. Another challenge is that the immune response to this disease is actually not completely understood, or I should say not deeply understood. And what I mean here is the human immune response to Lyme disease. Classical studies have used mice to model Lyme disease, but mice are actually reservoir hosts, which means that they're adapted to house Borrelia in the wild, and so they actually don't get sick like humans, and so they don't adequately recapitulate the human disease. And also laboratory strains of Borrelia differ from those that infect humans in the wild. So our strategy has been to conduct a deep dive into each individual, that is into each Lyme disease patient. And we are doing this by studying a cohort of Lyme disease patients and conducting systems level studies on them to track the immune response over time in these patients. That means that we are collecting information from the ground up on the genes, proteins, cells in these individuals and integrating it with clinical information that is derived from our clinical collaborators to really build a holistic picture of how the immune response develops and resolves over time and how changes in the immune system impact other organ systems over the course of Lyme disease. And we hope that these studies will help us build better immune-based biomarkers for Lyme disease that will help diagnose the disease hopefully early on than traditional diagnostics. And in the future, we hope that these studies will guide larger studies on post-treatment Lyme disease syndrome, which might help us understand why some patients never actually resolve the disease and get well post-antibiotic treatment. We certainly hope so, and this is currently an active area of research. Lyme disease can lead to long-term sequelae, sometimes in individuals who've been previously treated with antibiotics, in which case it is called post-treatment Lyme disease syndrome, and sometimes also in individuals who escape diagnosis, which is more classically called chronic Lyme, although this category can also include some other kinds of patients. Now, very interesting parallel between long-term Lyme and other autoimmune diseases such as SLE or multiple sclerosis is their very heterogeneous presentation in terms of the types of symptoms that they cause and the many different parts of the body that they can involve. So Lyme disease can also involve multiple organ systems such as the brain, the heart, and the joints. And so there are very interesting questions to ask in this area. Are there common networks, specifically immune networks, that underlie these different diseases, perhaps perturbation of a common set of pathways, or shared sets of autoantibodies that develop in these chronic diseases? These are still answers that we're seeking. Getting answers to these questions could have huge implications for potentially repurposing of drugs, which means taking drugs that are used for current autoimmune diseases and using them to treat long-term Lyme disease and vice versa. Over the past 10 years, immunology has transitioned from being a predominantly reductionist field to one with increased incorporation of systems-level approaches. And this has not only accelerated discovery, but also done so in an unbiased way, providing us a glimpse into immunity's black box. We've developed new hypotheses and new targets for diseases. However, we are still just at the beginning of exploring this black box, and a lot is still unknown. I anticipate that with conventional interdisciplinary barriers breaking and with the onset of programs that train students across disciplines whereby they learn both the biology and the computation, that these cutting-edge technologies and multi-omics will be brought to bear upon immunology at an accelerated pace. This has the potential to impact not only basic research, but also its translation into the clinic. In particular, conducting a deep dive into each individual across scales in conjunction with clinicians who know about the symptomology and the impact of the disease on the individual, we have the potential to develop personalized therapies and use big data to inform clinical recommendations.